Hello, everyone. I'm Ashima. I'm a graduate student in Dr. Chaudhary's research group. Today, I'll be presenting this poster on quantitative profiling of system level protease activities by mass spec cleavable signature ion based strategy. I'm going to start with giving a brief overview of what this poster is all about. So, in human body, proteases, which are nothing but enzymes that break down proteins, play a central role in various biochemical pathways which catalyze and regulate key biological events. A few examples of proteases are trypsin, pepsin, chymotrypsin, gluci, and so on. So these enzymes basically catalyze the proteolysis of proteins. Proteolysis is nothing but the breakage of peptide bond by hydrolysis reaction. So we are basically hydrolyzing the peptide bonds and breaking them apart so the proteins are converted into peptides. This is also called digestion, protein digestion. So these activities, these proteolytic activities can have destructive potentials since it can lead to various kind of diseases such as cancer, neurodegenerative diseases, some inflammatory conditions, cardiovascular diseases, and viral infections. Therefore, it's very crucial to regulate the protease activities by identifying the protease substrate and protease products under physiological conditions. And in recent years, mass spec based methods have come into picture to do so. So in this method, we are introducing a N-terminal tag, which basically tags the N-terminal of the protein. And by doing the bottom-up proteomics, which is nothing but the cleavage of proteins to peptides using a protease, can lead to the identification of various proteolytic products. So in here, I have synthesized a N-terminal tag, which basically reacts with this N-terminus of the protein. And since we have a lysine side chain, which also has a primary amine, so we have to first block the lysine chain so that that doesn't compete with the N-terminal tag. So first lysine undergoes guanidination, forming a homoarginine kind of product, which so now the primary amine of lysine is no more available to react with the N-terminal tag. So we can specifically label the N-terminus of the protein. So upon adding N-terminal reagent, we can tag the N-terminus of the protein. And since this N-terminal tag, since this tag has a MS cleavable bond, so it also reduces the data complexity. So from here, the proteins are digested into peptides followed by biotin evident affinity enrichment. So we can do this enrichment since the tag contains a biotin group and biotin has strong affinity with evidence, so we can specifically enrich our tagged molecules here, followed by the data analysis. So the use of this MS cleavable tag can reduce the data complexity of the mass spec analysis. Here I've shown the full MS of the N-term reagent. So it's nothing but the characterization that we have synthesized this N-term reagent using the solid phase peptide synthesis. So this 839.38 Dalton peak shows the molecular ion peak, the +1 charge state. And here I've shown the MS-MS spectra of N-term reagent, which characterize that the product has been formed, and 527.29 represents the signature ion fragment of the particular reagent. Then we moved on to tag this small peptide with the N-term reagent. So I've tagged RGDC peptide with the N-terminal reagent, and we have seen that the tagging has been observed with the sodiated +2 charge peak. So the, the peptide has been sodiated and we are seeing the peak of the same here. Moving on with this study, so the workflows goes like this. First, we start labeling the peptides, moving on to proteins. So my future goal for this study would be label the proteins and then move on to the complex biological samples such as cell lysate. Other than this qualitative study, we are also focusing on the quantitative study of protease products and substrates. So we are also planning to build the isotopic versions of the N-terminal reagent, which is isoterm 1 and isoterm 2. So here's the scheme for the same. So we're going to tag the proteins with the two different isobaric reagents, followed by proteolysis. And later on, we'll do the MSMS analysis, which can lead to the quantification of the proteolytic products, to which are important in the study of proteolytic activity. So we believe that this method is applicable to tag the N-terminal, and we can profile system-level protease activities using this technique. Thank you for listening.